Hello and welcome to Nikolai's genetics lessons, and today's problem is of the following DNA strands in combination with the complementary strands forming double helixes. 3 undergo a hyperchromic shift with melting temperature of about 42 Celsius. Which one has temperature of melting of 52 Celsius? First of all, let me explain what is hyperchromic shift is. Imagine that here is a test tube with a sample of the DNA. So here we have a DNA and we use laser beam. So this is going to be a source of our laser beam. Here is laser beam that goes through this test tube with DNA samples in it. And here we have a detector. So we know the intensity of the light, and we also can measure intensity of the laser beam at the detector site. Now imagine that this is double helix, and now imagine that these two strands of the DNA would separate, and when these two strands would separate, the absorbance of the light of the laser beam would increase substantially. And when we detect such phase transition in the light absorbance, we know that DNA separated. So now DNA, as we also say, melt. Now let's take a better look at these oligonucleotides. And in order to solve this problem, all you have to know that adenine base pair with thymine and guanine base pair with cytosine. And between guanine and cytosine we have triple bond, and between adenine and thymine we have double hydrogen bond. Take a look. For example, adenine would base pair with thymine and would produce double hydrogen bond, 2 hydrogen bonds here, and 2 hydrogen bonds here, 2 hydrogen bonds here, 3, 2, 3, 2, 3, and 2, and 2. Now let's find how many hydrogen bonds second oligonucleotide has. Of course, the more hydrogen bonds between two strands of the DNA, the stronger would be attraction between molecules, and the more energy we would need in order to separate these two strands, and the higher temperature of the melting is going to be. So again, let's count. 3 hydrogen bonds here, 2, 2, 2, 2, 2, and 2, 2, 2, and 3 here. Next, 2, 2, 2, 2, 2, 2, 3, 3, 3, and 2 here. And next, 2, 3, 2, 2, 3, 3, 3, and 2, 2, and 3. Now let's find how many hydrogen bonds first oligonucleotide has. Let's first find how many double hydrogen bonds. 1, 2, 3, 4, 5, 6, 7. So 14 hydrogen bonds here, plus 1, 2, 3. 3 times 3 is going to be 9. 14 plus 9 is going to be 23 hydrogen bonds here. Now let's count double hydrogen bonds. 1, 2, 3, 4, 5, 6, 7, 8 times 2 is going to be 16, plus 6 is going to be 22 hydrogen bonds here. Next, this oligonucleotide. 1, 2, 3, 4, 5, 6, 7 times 2 is going to be 14, plus 9 is going to be 23. So 23 hydrogen bonds here. And the last oligonucleotide. First, how many double bonds we have? 1, 2, 3, 4, 5. So 10 hydrogen bonds here, plus triple hydrogen bonds. 1, 2, 3, 4, 5. So here we have 25 hydrogen bonds. Now we are told that 3 out of 4 has melting temperature of 42 degrees of Celsius. So we can group these 3 oligonucleotides together, and the one with highest number of hydrogen bonds would have temperature of 52 degrees of Celsius, and this is going to be our answer. So this is have to be this oligonucleotide because it has more hydrogen bonds. And here we would have a temperature of the 42 degrees of Celsius. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. And see you in the next video. Goodbye.